I want to kick off this discussion with the most basic question to both of you. Can you help us understand what precision or personalized medicine is? Maybe they're both one and the same, or there's a difference. And we'll start with Dr. Collisson, and then Dr. Gilbert, you can chime in.
Sure. I can start. I think of you mentioned precision medicine and personalized medicine, and I think they're a spectrum of the same phenomenon. Precision medicine, I think, refers to treating the molecular underpinning of a disease, and what Dr. Gilbert and I work on mostly is cancer, and what that means most of the time is a mutant gene or a mutant gene product. And so precision means that we're looking for chemical matter, most often drugs, sometimes other treatments like cells or other proteins that attack the underlying cause precisely. So in contrast to chemicals that kill many different kinds of cells, we would like to be precise in what we do with the treatments we give, and that is called precision medicine. And I think personalized medicine is along that spectrum, but folding in the diagnostic angle, which is increasingly important in the clinic. It was fine years ago to sequence one genome over 20 different centers over 20 years with close to $1 billion. Now our challenge is to do that in one human being in 48 to 72 hours so that we can figure out what mutations that patient's tumor harbors in enough time to get the treatment on board when it's needed. So I think precision medicine is the overarching goal, and personalized medicine is the reduction to practice with the diagnostics in the clinic.
I think, from my perspective, I think what cancer biologists do is more precision medicine focused, and what clinician scientists do is both, right?
Yeah.
So we think more about precision medicine on the level of targeting specific mutations or specific tumor genotypes or tumor genetic spectrums, and then the implementation is an enormous challenge to something that I'm frankly quite blind to.
So what we're saying is that precision medicine is an approach to the care of cancer, since we're talking about cancer here, in which we are trying to match probably the best therapy with the optimal patient. And for this, we look at all possible information about the patient and the disease. Is that right?
Eric may disagree with, or Dr. Collisson may disagree with me, but I think precision medicine is a combination of science and medicine, and personalized medicine is medicine. I don't know. Would you simplify it that far?
Yeah. No, I think that's well said. That the precision we seek to gain is really at the molecular level in cancer. That we want to know the underlying cause, and that's just essentially a scientific enterprise of testing hypotheses. Personalized medicine is you can't always test a hypothesis in the clinic. You've got to practice medicine, and that's both art and science. And personalized medicine falls in that spectrum.
Yeah, thank you, Doctor. So I'll just ask one more question on the basics before we actually address the topic that we're discussing today, whether am I eligible for precision medicine. So, Dr. Collisson, it will be great if you can very briefly, this is for the benefit of the audience so they know what we're talking about here, very briefly summarize the kind of therapies that are presently available under the precision medicine umbrella for cancer. Just a brief idea of some terms that probably the listeners are quite familiar with, but would like to hear.
Sure. I'll give 3 examples, and they'll be brief. They all involve therapeutics, because that's usually, if cancer's left alone, unless you treat it with a therapeutic, then it usually ends in death. Not always, but quite often. So we're interested in treating cancer because the results of not doing so are lethal. So the three therapies can be broken down into basically small molecules. So these are drugs like antibiotics or aspirin that many patients are familiar with. They come in pills or in infusions. And these drugs often bind to proteins that we know drive cancer proliferation. So this is the equivalent of a broken accelerator that cannot be lifted off the car floor, and the cancer cell is essentially floored and always accelerating. So if you can design a piece of chemical matter to fit into that accelerator, you can pull it off the floor and get the acceleration to stop. And there are great examples of that Dr. Gilbert uses in his lab. Imatinib for CML, for leukemia, is an example. We have many other examples in melanoma and lung cancer that we work on as well. A second example would be a marker, like a protein on a cancer cell. So it may not drive the cancer cell, but it flags to the therapy that this is a cancer cell. And there we've been able to attack those markers with antibodies like Herceptin or rituximab. These are antibodies that attack the cancer cell by the identification of this protein on the cancer cell itself, and then the cancer cell gets killed by the immune system. Or increasingly, we skip the middleman and we make the immune cells ourselves. And this is exciting work happening at UCSF by some of our colleagues, where they actually engineer the patient's own immune cell to attack the cancer through a similar recognition of this cancer-specific epitope. And their CD19 and other liquid cancers have been successful early examples, but we expect more. So the idea is always to achieve a therapeutic index, meaning that the cancer cell dies, the normal cells don't, based on a molecular lever, meaning that the cancer cells have this thing, the scientists have discovered it and studied it, and then a pharmacologist or other person has figured out how to make a therapy to block it.
Mm-hmm. Thank you, Dr. Collisson. Looks like we have already started getting a few questions in my email. So this one I think I'm going to take now. It's a person wants to know what cancers can be treated with precision medicine. I know you just mentioned leukemia, melanoma, and lymphoma. So what else? What are the other cancers that can be treated with precision medicine? This person wants to know.
Sure, great question. Well, I think at the level of cancer, we're increasingly trying to do the first step in precision medicine that we talked about, at least in personalized medicine, which is diagnosis. We want to achieve precision diagnosis so that we don't get into the scenario where we don't know what this cancer is, we don't know what's driving it, we don't know what's going on. That's obviously not a good starting point. So I will say all cancers, and maybe Dr. Gilbert will disagree, but all cancers are amenable to a precision approach to diagnostics. And that today, I think, includes imaging of the tumor using PET scanning and CT and MRI scanning. These are precision imaging approaches, as well as genomic characterization of all tumors. All tumors have DNA. All tumors have at least one mutation in them, and figuring out the mutations that drive the cancer is a key first step. Then the treatment is the next step, and we have FDA-approved, quote, precision treatments in many diseases. Certainly the biggest cancer killers, lung cancer, breast cancer, and colon cancer, all have precision approaches in various degrees. There's still some pretty egregious omissions on that list. Pancreas cancer that we work on a lot in the lab, hepatocellular cancer, these are cancers that kill millions of people worldwide and for which we haven't really made a lot of progress on the precision medicine front yet, although that's changing. Thank
you, Dr. Collisson. My next question is Dr. Gilbert. Dr. Gilbert, what is the difference between genetic and genomic testing and sequencing? The reason why I'm asking this question is when we were just preparing for this talk and getting some feedback from patient advocates, there seemed to be a big confusion around the difference between these two. It would be great if you could just clarify this.
Yeah, I think many people do use them interchangeably, and there are more specific definitions that can be applied. But the confusion in part relates to the fact that as we diagnose cancer, our methods for diagnosing the underlying alterations have changed over the years. And so my definition for this is for genetic testing, you can be looking at a single gene or all genes. And for genomics, sort of by definition, you're looking at thousands of genes or all genes encoded by the genome. And so older tests in the late 90s would, for example, look at a single gene that was known to be associated with cancer and ask, is that gene mutated in an individual patient or a cohort of patients? Where currently we're moving towards not looking at single genes in patient samples, but instead looking at either a large fraction of the genome or the whole genome. So if you're at a cutting-edge cancer institute, often as you enter, you can be given the option to have a test run on all genes known to be associated with cancer, for example. There are companies that do this, like Foundation Medicine. And so we may, because of our change in methodologies, we're losing the what we would have historically defined as genetic medicine, and everything's becoming genomic medicine in my estimation.
Yeah, I would agree. I would add another possible distinction between the two terms, and I agree with Luke that they're used, I don't want to say sloppily, but these are new terms. The word genomic didn't exist a decade ago, so I think we're trying to fit where it fits. One thing we use in cancer, genetics often refers to the genes you came with, meaning your mom's and your dad's. So germline genetics is a term that's often used, and these are the heritable components of cancer, which we know are many, whereas genomics may refer to the somatic changes, the changes that the cigarettes or the UV rays or the bad luck that your cells had after birth and resulted in a spectrum of mutations that you were not born with. So some people use the term genomics in cancer genomics to say, oh, these are somatic changes. I don't know if this word's in the dictionary yet, actually. Maybe it is.
I'm not sure. Yeah, no, that's well said. That's a nice way of looking at it.
So when we talk about genetic testing, most of the time we're talking about genes that have been inherited from family members, and when we talk about genomic testing, we are talking about our own tumor and somatic changes from our tumor.
Yeah, so ideally, to add to that, then your genetics doesn't change over the course of your life, where the genomics of a tumor can change between your normal cells versus your tumor cells, but also during the course of treatment, a tumor can change, and so you would want to get genomic profiling ideally throughout the course of tumor treatment.
Mm-hmm. So we just had this question just came in, I think, based on what we're just talking. The question is, will genomic sequencing show if I have a hereditary risk for cancer?
Yeah, it's a great question. You, like most tests, you only know what you test, and so some of these panels, they interrogate 50 genes or 90 genes. At UCSF, our in-house panel looks at 500 genes, and there's more than 20,000. I think it's now 28,000 genes in the genome. And we don't sequence all those, so. But you do inherit all of those times 2 from your parents. So we think a large amount of cancer risk is inherited, meaning up to 50% in some cancers, and certainly 10% in most common cancers. There's a familial risk component, meaning you're born with it. The genetic tests that we do today identify the major risk genes, but don't identify every gene that impacts risk. So we encourage people who have a family history, meaning mother, father, brother, or sister, to undergo germline testing in most heritable cancers, and those include pancreas, breast, melanoma in some cases, other colon cancer in other cases. So in those patients, we always recommend that you provide a saliva or a blood sample, send it off, see what mutations you have, and if something's found that is suspicious for the cancer that you have, then we work out from there. And if there's the appropriate consent and counseling in place, we talk to the brother, we talk to the mother, we talk to the children about their, usually 50%, but not always, 50% risk of inheriting that allele from you, if that makes sense.
Yes, that does. Dr. Collisson, maybe we could just talk a little bit about who is a good candidate to get tested and what exactly do they get tested for, and what might you hope would come out of that test?
I'm assuming you mean a germline genetic test, an inheritance test. Yeah. So so this is a really the Wild West today. I think as recently as about 3 years ago, the answer would have been very easy. If you have breast cancer or ovarian cancer or colon cancer, and you had a family history, then or your tumor looked a certain way under the microscope, in the case of colon cancer, then you should get genetic testing. That's still true and unassailable and absolutely true. We've now included other cancers under that rubric, prostate cancer, pancreas cancer, others in which we know that there are inherited mutations. So I would say any patient with metastatic prostate cancer, any patient with pancreatic cancer, any patient under 50 with colorectal cancer, any patient with ovarian cancer, and patients under, I believe, 50 with breast cancer of certain kinds should get germline testing. Less so for diseases like lung cancer and some brain cancers. But now we're also seeing the direct-to-consumer movement pioneered by Silicon Valley and other places where we're both getting, are you inherited, are you related to Aunt Josephine in Pittsburgh, so a relatedness test to kind of build interesting networks of long-lost cousins, but also spontaneously discovering deleterious cancer genes at the same time. And this is a bit of an ethical struggle of how to deal with these. If this patient just wanted to know, is she 20% Irish like mom says, or is she actually from Germany, but actually finds out that she has a 50% chance of getting breast cancer, that's not really what she signed up for. So that's difficult to know how to deal with that. And then a third scenario is there's a bunch of companies coming through that are proposing to build blood tests to actually find cancer before it's clinically evident, and those look very encouraging, but are definitely too early for prime time. But this is a rapidly moving area.
Thank you, Dr. So I believe when we last spoke, we discussed how it's definitely not just about the information. It's what you do with that information. It's about how appropriate treatment is. And when we are especially not sure if the gene mutation will respond or will not respond. So, doctor, do you have to address the situation about appropriateness of selecting treatments? And how do you address this with your patients?
Well, I try to look at first, clinical evidence. Has an experiment been done in patients like this patient with this drug? That's the highest level evidence. But I think there's, unfortunately, the vast majority of the diseases in the cancer clinic today, we don't have good treatments for. And I increasingly look to people like Dr. Gilbert to tell us, what are we not thinking of here in the clinic. I'll let him explain how he finds some new therapies for cancers, and actually how he figures out how certain drugs we use all the time actually work.
Yeah, so I think as a background piece of information, and Dr. Collisson alluded to this, is we only defined what the genes are in the human genome at around 2001. So it's been less than 20 years since we've even known for a single genome what was encoded in our genome. Since then, we've come a long way, due in part to advances in technology, and we've now sequenced hundreds of thousands of healthy individuals from across the globe, which defines what the natural genetic variation in the human population is. We've also looked at many hundreds to thousands of genomes from individual diseases and defined what the genetic alterations are that are associated with individual diseases, not just cancer, Crohn's disease, lupus, all kinds of neurodegenerative diseases. In the context of cancer, we've sequenced hundreds to thousands of genomes from individual cancers, and that number continues to grow. So for any common cancer, we now have a very advanced draft genome of the mutations that are associated with disease at diagnosis. For, I'd say, a fraction of those changes, we as a scientific and medical community have built precision drugs. One of the more recent examples of this is a gene that is called KRAS that's mutated very commonly in lung cancer, pancreatic cancer, and colon cancer, as well as sporadically in other cancers like leukemias. And it's been really a holy grail of thinking of new ways to target this gene. We know this gene drives tumor progression, and it drives poor response to conventional therapies. And so our colleagues at UCSF a couple of years ago came up with a really innovative way to target this protein, and that's progressed through startups and big pharma to a series of molecules that were presented this year at ASCO, which is the main clinical trial conference in the U.S. for cancer therapy, with Amgen announcing that they have pretty dramatic responses in using this molecule to treat lung cancer patients that have a specific mutation. Despite the promise of these approaches, we have a long way still to go. So for I'd say most of the genetic alterations that we see in an individual patient, we don't have specific precision therapies to target these mutations, and even worse, we often don't even have an advanced understanding of what these genes do or how these genes work together with other changes we see in tumors to drive complex biology within an individual patient. And so there's much to do, and there are many people working very hard on this. I mean, this is the fundamental challenge in cancer biology at this point.
Yeah.
Dr. Gilbert, but isn't it true that there are reports that confirm that the majority of individuals who have contributed their DNA to genomics research are Caucasians, and therefore minorities underrepresented in the genome database? Can you comment on this as well?
Yeah, to my understanding, that's absolutely true. The majority of the genomic information we have for cancer is derived from the U.S. or from Europe or to some degree from Japan and China, but that doesn't cover the global spectrum of genetic diversity in humans. We have intentionally collected normal human genome information from across the globe, so we have that part in place. But I think it for sure in the context of many cancers, we don't understand how the mutations that are associated with liver cancer in Southeast Asia are similar or different from the mutations associated with liver cancer in North America. And one of the examples, I bring this up as an example because there are global differences that don't relate necessarily to race, but instead even to diet. So there are known toxins associated with peanuts in Southeast Asia that drive mutations in liver cancer, which gives those people in that region of the world a different mutational profile than what we have in the U.S., where we have liver cancers that are more associated with obesity and alcohol consumption and other influences. So definitely an important, you don't want to, we need like your whole podcast is pointing out, we need both a precision and a personalized approach here, and that applies to each individual's family history as well as their personal life history.
You know, that's one of the shortcomings of, for all the excitement around genomics that we talked about, putting things into the DNA sequencing machine and getting lots of data. That's all very exciting, but what we often forget what we don't gain from that, and what we don't fully get out of the sequencing machine is, you know, where did this patient come from? Is this patient born in the U.S.? Does this patient have a virus, you know, that she had as a child? Is this a smoker or a non-smoker? What are the effects of environmental toxins or other practices? And so I think increasingly we're trying to get these well-annotated data sets that go beyond just genomics. So it's putting that cancer genome from that patient in the setting of who she is and how did she grow up and what was her diet like and her exercise like. And it's only with that complete picture that we're able to really educate the next patient. You know, in patients like you with this mutation, we found that a high-fat diet is especially deleterious, for example. We can't make that give that advice if we don't have the dietary information with the genomic information. And that's a major challenge. We're somewhat handcuffed in the medical centers today, unfortunately, due to the privacy with which we all guard our medical information. And that's probably appropriate, certainly for diseases with more social stigma to them, but I think in metastatic cancer, there's a group of us who are trying to loosen those privacy rules, patients being at the front of this charge, and trying to liberate their data for study on a broader scale, to put their genomics in context with who they actually are and get it out there. So that's something we're trying to do more of in the cancer community.
Thank you.